In this example, transcription will be occurring from left to right direction. Our initial RNA will be coming in from a 5' to 3' direction. Because we know that our RNA is 5' to 3', we can label our template strand, the complementary strands of RNA. Down here is 3' to match up, 3' to 5' complementary, and over here. Our coding strand will be the opposite and will be matching with our RNA. Before transcription can actually occur, we must first have RNA polymerase bind to our DNA. The region where RNA polymerase binds is called the promoter region. RNA polymerase will be attracted to these things called consensus sequences, labeled here as -10 and -35. -10 relates to the TATA box, which is a code recognized by RNA polymerase, which attracts it and will want to bind. Also, the -35 serves the same function. Our +1 signifies where transcription actually begins occurring. However, before we begin this, I want to make a note of our core enzyme and sigma factor, which equal our hollow enzyme, which also aid with the binding of RNA polymerase to thus begin transcription to occur. Now that RNA polymerase is attracted to our DNA and bound via our hollow enzyme, to actually begin transcription, our sigma factor must be released and broken off to now facilitate and carry through transcription to go down the chain. Now that RNA polymerase is bound, our transcription can occur. So it goes past the promoter region into our RNA coding region being built, and our DNA is transcribing into our RNA, messenger RNA, until eventually it will reach a termination sequence. Our RNA strand can break off from the DNA strand when an inverted repeat occurs in our two DNA strands. So for example, if our upper strand had the AGGC form and our lower strand had a GCCT, this is called an inverted repeat, which would lead, as it goes through transcription, it would eventually lead to an inverted hairpin type formation with the RNA tail end going out with a bunch of U's, so U, and U's, go with adenines, and it's a very weak bond. It also diminishes the hydrophobic effect. Thus, our RNA strand would split from the DNA strand, leading to our separation in our RNA and DNA. So we want to hypothesize the consequences transcription would have if our -10 were to be deleted from our DNA. So we know that the -10 attracts our RNA polymerase. If this were to get deleted, our RNA polymerase would have trouble ever binding to our DNA. Thus, transcription would never be able to occur. Thus, our sugar that we want to form would be unable to be made.